Hi, this work is titled Sex Differences in Alzheimer's Disease, a meta-analysis of microRNA studies. It consisted on a compilation of microRNA expression studies focused on AD that also included sex information, in order to analyze this data including the biological sex in the contrasts, as well as the disease condition. Alzheimer's disease, AD, is currently the most frequent form of dementia, and it causes neurodegeneration and brain atrophy. AD shows differences between biological sexes in several aspects of its development. Thus, taking sex into consideration when studying the disease could allow for the discovery of new sex-specific biomarkers. MicroRNA molecules inhibit the expression of their targets. The alteration of this mechanism of regulation has been linked to the AD pathogenesis. In this work, we aim to identify potential microRNA AD markers by integrating the results of differential expression analysis of microRNA in AD, in which we also contrast the biological sex variable. The methodology followed consists on a pipeline in which we conducted a systematic review of the microRNA expression studies centered in AD that also included the biological sex of the samples. Afterwards, we explored the data of these studies to evaluate the distribution of expression values and the distribution of samples among conditions. We also evaluated the annotation used for microRNAs in order to update all studies into a common annotation. The following steps was a differential expression analysis of the selected studies using the Lima or Lima plus Boom methodology in microarray and RNA-seq studies, respectively. We designed contrasts that involved the sex as well as the condition of the patients. Finally, we carried out a random effects meta-analysis to integrate the differential expression analysis of microRNAs present in more than one study. Regarding the results, we got a final selection of 6 studies that met the selection criteria. After setting a common annotation for the microRNAs included in each study, we evaluated how many of them were common to several studies. The upset plot represents the intersection of common microRNAs. With regards to the differential expression analysis, a contrast of the effect of AD in women against that effect in men returned no significantly altered microRNAs. However, contrast of exclusively women or men returned altered microRNAs. The meta-analysis of such results allowed for the identification of consensually under or overexpressed microRNAs. Some of those microRNAs were not detected in the integration that didn't consider the sex of the patients and could be potential new AD biomarkers. Finally, we arrived to several conclusions in the development of this work. It is important to adhere to fair guidelines to publish accessible, reusable data that can be integrated with meta-analysis techniques. Meta-analysis allowed for the identification of potential microRNA biomarkers altered in expression studies for AD patients when considering the sex information. And finally, the research on the role that microRNAs play in AD as well as the sex differences are still a challenge. Standardization of microRNA libraries and consideration of the biological sex of the patients may be key for understanding this problematic disease.